So welcome to this first Protist eDNA bioinformatics workshop. That's true, when we organized this small meeting, we didn't expect that much people that would be interested by this topic. So it's very great for a first attempt to organize this kind of Protist workshop. And it just shows that there is a strong interest by the community by such events. So I would like to thank of course, Guillaume and Enrique to join this workshop. Each have different specialties and can bring some skills about these DNA concepts. So first things, I would like to give you a big disclaimer. This workshop is only 1.5 days long, so you will not become an expert in this field of research in just 2 days. And this field of research is constantly evolving. And the things that are true today, maybe in 2 years, it will change. And there is also different approaches and methods that are scientifically correct and valid. So we will bring you our vision of Protist eDNA metabarcoding studies, but there is other way to analyze your data set also that are correct and valid. So we will give you an overview based on our 7 years of experience. We send it to you a short survey, and here is the results. So as you can see, for the field of research, there is different interest with biomonitoring, biogeography, macroecology, diversity, and some people in paleoecology, and also other kind of field of research. The same for the different environments. A lot of people, more than half of the people in this conference are interested by marine environments, but you have also some people that are studying soils, fresh water, and all different kind of environments. And about the organisms that you are studying, half of the people are studying the whole diversity of eukaryotes and fungi and bacteria. There is a huge community of Foraminifera, thanks to friends that send you to the link to this workshop. Also about Testate amoeba, cyates, diatoms, and other kind of organisms. So of course, we will not be able to cover every individual case. So why study Protist eDNA? I just want to start by giving you a few examples of studies that use eDNA to answer some questions. So you can use eDNA to see the diversity across the different ecosystems. The first study here we compare marine organisms, fresh water and soils. And basically we can highlight the hotspot of each group according to the main ecosystems and see the differences of community composition in these systems. The second study, Jens Bönig and his team just studies the freshwater community in lakes, but in the European scales, they did an amazing data set to study the relation in the structure of community composition. You can also see with eDNA metabarcoding studies the role of protists, for example, in the carbon cycle. And the contribution of protists in this cycle are really huge, and it was quite unexpected. For example, in peatland, it's more than 10% of the carbon that are uptaken by microorganisms. And also, there is the microtrophies that are really important. And you can assess this kind of question with eDNA also. You can also use eDNA studies to see, for example, the importance of parasites in the ecosystems. And it was also unexpected that there is a huge amount of these parasites that contribute in the network food webs. So this is a few examples of eDNA studies that you can do. So the application of Protist eDNA, you can study the taxonomic diversity, you can do the ecology studies, biogeographical studies, functional diversity, biomonitoring, and there is also different applications that you can think. So it's a very broad area of things that you can do with this Protist eDNA. Just a few words about Protist diversity for the people that are not used to work with the whole diversity. The diversity is really, really huge. And in this graph, you can see the diversity of plants, fungi, and animals, and all the rest are Protist diversity. And if you look on the Foram, for example, the Foram are here. And you can imagine that each of these names are as diverse as Foraminifera. And of course, you will not study the same way Foraminifera or Testate amoebae or C8 or parasites. Each of these kind of organism request different approaches, different methods to collect them and analyze them. So now, after this short introduction, I will provide you some eDNA concept from the sampling to the sequencing. So first, an idea about the eDNA workflow. So first you will do the sampling. So you can sample any kind of sort of materials. Then you will prepare your sample in the lab. So you will do the DNA extraction, the PCR, and the sequencing. Then you will do the bioinformatics. You will filter your reads, do the clustering, the assignation. And finally, you can do the analysis and statistics. So today we will focus on the sampling and the preparation of the samples in the lab. And tomorrow we will cover the bioinformatics. I will do the first part about the filtering and the clustering. And then Enrique will explain how to assign well your OTU ASV to a known species. And with Guillaume, you will learn a bit more about statistics with R and which kind of analysis you can do with R. So first thing, when you want to do an eDNA study, you should have a plan. You should plan your studies. So you have different things to think about. So of course, there is advantages of eDNA studies, and this kind of slide you will see in all the conferences. They will always say, oh, the price is very low, the time is it's very easy to do this kind of analysis and have the whole diversity of every group. So that's true, only if you have a very strong reference database. And if the group is well known. If you work in a group that are not well known, it will be much more complicated and it will take you much more longer time to assess this kind of diversity. And when you are planning to do eDNA studies, you will need to do a budget, like in every kind of experiment. So it's very easy for the consumable and the analysis for the project. But often people forget to plan the human resources to analyze the data sets because it's not straightforward and it takes a lot of time. So you should really think when you design your experiment, a big part of human resources, or you will not be able to get the results out of your raw data sets. So just a few words about experimental design. You need to think about the number of samples, the number of replicates you want to have in your analysis, the type of the samples, of course, the preservation of the raw materials, the DNA extraction kits that you will use for your eDNA studies, the primers, if you want to use universal or specific primers in your experiments, the PCR and the library preparation, the sequencing platform that you will use, and have an idea about the bioinformatics you want to perform on your data set, the assignation also, and the statistics. And of course, all of these points are really important because if you change something here, it will influence your results. So having a good idea about all these points before starting your experiment will save you a lot of time at the end. So we will go through all these points during this presentation. And if you want to discuss more about experiment planning, project writing, or budget, we will have a breakout room free. And it's there that you can discuss with Enrique all these different concepts that are really, really important when you are planning eDNA studies. Once you have planned your studies, you will have some field work. So a few tips about the field works. So first, the number of samples and replicates. It exists really different strategy depending on the studied environments. So from my own experience, I worked with sphagnum peatland and the littoral zone. And these two environments are really, really different. And for the first one, you have a huge stability for sphagnum peatland, but for the littoral zone, it's totally different and it's very heterogeneous, and it's totally different dynamisms between these two ecosystems. So if the stability is very high, you need less samples to cover the whole diversity. So you can think to have more sites. But in the littoral zone, as it's very difficult to catch the whole diversity, you should think to increase the number of replicates, so you will have less sites. So it's up to you to find the right threshold between the number of samples you will have, the number of replicates, and the number of sites. And it really depends on the ecosystem that you want to study. So you need to think how much sample I need to cover the whole and to catch the whole diversity. Of course, after that, the type of sample, there is different strategy depending on the studied environments. If you study coarse, marine or freshwater, you will collect a sample differently than plankton, or just plant, or soils, or air, or any kind of material where you can extract the DNA. And for all this kind of environments, you should pick the right strategy for your target groups. For example, in soils, if you are studying nematodes, you will not collect the same way the soils than if you are studying C8s, for example. So you should select the best method to sample your targeted groups. So the preservation of the raw material. So the best practice is to extract the DNA as soon as possible. Really, it's a way to obtain the best quality of DNA and have the best results. But of course, due to field constraints, it's really sometimes not possible to extract the DNA rapidly. So it exists several options to store temporarily your sample. The first one is a preservation buffer, like the lifeguard. So this kind of preservation buffer have a very high efficiency, but you cannot really use this buffer to preserve a huge quantity of samples. Why? Because the cost is very, very high. It's about $2,000 for 1 liter of liquids. And with 1 liter, you can preserve like 1,000 sample of 1 gram. So it's if you want to study soils and you need to extract DNA from 10 grams, it's not the best solution. Another solution is to freeze your sample at -20 or -80. The efficiency is depending on the group you want to study. For some group, it's worked quite well. Like for Foram, we have some great results with frozen sample. But for other groups, sometimes it doesn't work very well. I had quite a bad experience with the V4 region of the 18S on a soil sample where the sample were frozen. So it really depends on the groups. And with that, the sample quantity is not an issue anymore because you just need a freezer and the cost is very low. If you want to freeze your sample, you should freeze very strongly when you collect your samples. And this is a way to just start the community to evolve in your sample, and especially for the small flagellate and the small groups like this, that can really evolve really fast in your samples. Once you have done with field work, you have the molecular laboratory work. So in this part, I will show you an overview of the different steps. So you will have your raw samples. The first things will be to extract the DNA. The second things will be to do your PCR. So here is metabarcoding. What we we will do with PCR. We will do 3 times PCR per eDNA extraction. And then we will pool this 3 PCR product in a single sample. This way, we will avoid PCR, or no, not avoid, reduce the impact of PCR bias. Once you have done with the PCR, you will quantify the quantity of DNA you have in your sample, and you will pool the sample according to the libraries that you want to do in the process. You will prepare the library. A library is the way you need to prepare your sample before the sequencing, and it's depending on the sequencing platform. And at the end, you will sequence your samples. So this is just a general overview of what you want to do, but depending which sequencing platform, for example, you can just send your DNA extraction and they will do all the rest of the steps. But it's just to provide you an overview of what you need to do to sequence your samples. So if you want to discuss the field work and the laboratory work, it will be in the breakout room number 1, and I will discuss these topics with you. Now about the DNA extraction kit. Depending on which kind of DNA extraction kit you will use, you will have different community profiles. So it's very important to select one DNA extraction kits that are really well adapted to your raw materials. For example, if you are studying soils, in soils there is a lot of inhibitors of PCR. So there is some kits that are designed to remove most of this inhibitor PCR. So first choose your kit according to your raw sample, and then during the DNA extraction procedure, you have steps when you need to do the bead beating. And the bead beating also should be adapted to your targeted organisms. Of course, if you have some organisms like forams, they are living in a test, so maybe you should increase the bead beating time to crush the test of this organism and release the DNA. So this kind of steps should be investigated before doing your PCR, your DNA extraction. So you need to choose the same kit of DNA extraction and use exactly the same protocol for all your samples. If you change the kit in the middle of your experiment, you will introduce some bias in your data set. So it's very not good to change in the middle of your procedure. And right now with the pandemic, sometimes it's very difficult to buy this kind of kit. So the best thing is to buy them before starting your experiment, so you will be sure that you have all your kits ready for the DNA extraction before starting the eDNA studies. Here, I just want to give you some first results we got to compare a kit, the 10-gram DNA extraction kit with the 0.5-gram DNA extraction kit. And we focus this study on Foraminifera. And as you can see, both kits will provide you different outputs. And you cannot find really the same ASV or OTUs using both kits. So we should now investigate why we have such a result, because it's an issue. And maybe use these 2 kits to cover the whole diversity. We don't know. We should really check more what kind of organisms are extracted using these 2 methods. Then, once you have done with your DNA extraction, you will need to choose your primers that you want to use to amplify your organism. So you have 2 different strategies. You can choose to use a universal primer like the V9 or V4 region of the 18S, or the ITS or the 16S for bacteria. So if you choose to have a universal primer, it's great because you have a good reference database with a lot of information, but you will not have a lot of phylogenetic information. So you cannot really assign to species level because you don't have enough information about that. So I just put a plus and a minus because it will depend on the groups that you want to study. Some groups will have better phylogenetic information, and for the other one, not so much. And of course, with the universal primers, right now there is a lot of studies that are published where you can find a lot of information. But of course, the lack of phylogenetic information is an issue. So it's why now we try to develop more specific primer applied to eDNA metabarcoding study, like the CO1 or a MATCA, NAD, or some specific primer in the 18S. So it's very great because you will increase a lot the phylogenetic information, but you will have a very bad reference database, and you will obtain a lot of environmental clades, and you will don't know exactly what are living in your samples. And of course, there is not so much studies using this specific primer, so you need to develop the whole methods. And just a few words about that. The OTU phylotypes that you will get with your bioinformatics is not equal to species, especially for universal primers. If you are using some specific primers, maybe you can go to that level, but be really careful when you are writing your papers, especially if you are using V9 or V4. Even if you have 100% of similarity between your ASV and your reference database, you don't have enough phylogenetic information to be sure this is this species. So be careful with that. And also the abundance of reads you will get is not equal of the abundance of individuals. So there's a lot of studies that discussing these issues. And for some groups, you can correct the number of reads by the biovolumes to try to obtain some relation, but it's a very case by case and it depends on the group. So be careful when you interpret your data sets. Then once you have your primer, you need to design the barcodes. And the barcodes, there is a number of studies that provide you some barcodes that you will attach to your primers, and these barcodes are used to separate, discriminate your different samples. And in this figure, in below the number 4, you can see in the green color, it's your primers, and in the red color, it's the tag of your sample, and in blue color is the adaptators. And the adaptators is what the sequencing platform will need to sequence your umbilical. So you need to have a great barcode, because even if you design some barcodes, depending on the primer you will use, if you look on the box plot, there is an orange one. That means this particular barcode makes some issues when you sequence them. So there is some specific issues regarding eDNA high throughput sequencing, but if you design well your primer by adding the right barcode depending on your primer sequence and some spacer, you can prevent this error. Sorry. So there is also a question when you do some Illumina sequencing, there is a tag that can jump from one sample to another and can make some error in your sequencing. So this is a topic that are also discussed in this paper. And about the quality of your sequence, if you don't have a lot of species, the sequence will be really similar between them. And having a high similarity will reduce the quality of your sequencing. So this is why we add now some spacer in our primers. So once you have done this step, you will need to select a sequencing platform. So you have an Illumina platform, a Pacific Bioscience PacBio platform, or Nanopore. But for metabarcoding, the first thing we have to check is the size of the reads that you will obtain. So often it's 2 times 250 burpace or 300 burpace. But be careful, because the size that you need to have should include the adaptators, the tag, the primers, and like 10 nucleotides for the overlap between your two, between R1 and R2. Then you should check the quality of the output, and it's very important to have a good quality, very good quality, because if you have some error during your sequencing, it's not good because it would create potentially some new ASV and new diversity made just by error. And finally, the last thing you have to check is the number of reads you will have in your sequencing. And the number of reads will depend on the quantity of ASV OTUs you will get to cover the whole diversity. For example, now with the V4 region of the 18S, with 50,000 reads, we think we cover quite well the diversity. So actually, the best consensus is Illumina MySec platform, but all this kind of platform evolve really fast. The technology, it's really impressive how fast it evolves, and it can change in the future. So just be careful of that. So if you want to discuss more about libraries, preparation, sequencing, platform, tag jumpings, you should discuss with Guillaume and go to the breakout room too. He's really the specialist of that. I'm not a very specialist, so he will explain really better than me this kind of aspects. So just a few words about the output files that you will get from the sequencing platform. So you will obtain 1 or 2 if you sequence per head, FASTQ file that will contain all your samples. The name of your FASTQ file will be like the name of your sample r1.fq and the name of your sample r2.fq. And each individual read is represented by 4 lines. So this is what it looks like when you open on a notepad in your computer. So the first line is the ID of the read, and it always starts with a @ Then you have the sequence of the reads. The 4 line is a plus, and it's useless, basically. And the 4th line is the quality of each nucleotide that you will use to assess the overall quality of your sequencing dataset. Of course, I provide you an overview of the good practices, but you all have some existing datasets that don't use all the methods and all the tips that I have provided you. And they are just waiting in our servers to someone to analyze them. And it's often the role of a master or PhD student or a postdoc. So even if these data sets were not designed with the best practices, they are really, really very valuable. So you still can analyze them and publish the data, of course. But you should ask and discuss the strength and the weaknesses of the data set with the people that sampled and did the laboratory work. And it's very important to know this strength and limitation of your data set to avoid misinterpreting your results. I can give you an example. For example, if you don't do a triplicates of PCR per eDNA extraction, you will sometimes have an ASV that will just explode in your results. But it's just maybe a PCR bias. So by knowing how the data set will be done, it will really save you sometimes and avoid you to overinterpret your results. Just a few words about the practical case from tomorrow. Tomorrow, we will work on data sets that come from Switzerland and France. And we sampled 5 different peatlands. And in these peatlands, we sampled 5 different microhabitats, the hummock, lawn, pool, forest, and transition. We got 65 different samples, and we sequenced the V9 region of the 18S RNA gene. So we use a universal marker for this data set. Here you have a picture of what it looks like. So you can see in the peatland, you have some pool, alone, so it's a flat region of the peatland, and some hummock, different sphagnum species in this microhabitat, and we investigate which kind of microeukaryote we got in these environments. So we have 5 microhabitats, and of course we just have a few environmental variables like total nitrogen, total carbon, and water table depth. That's a level of it's a kind of related to the humidity of the sample and the pH. So we are nearly at the end of this presentation. So here is some take-home messages. So the experimental design step is really crucial. You should think about how you will perform your DNA studies. And your workflow must be adapted to your targeted organism. There is no standardization where you can have the whole diversity. It just doesn't exist. And your data set must be homogeneous, and so you don't change the methods in the middle of the experiments. And with these few points, you will have a really good quality data set and great results. So I thank you for your attention. We still have 15 minutes for questions before we can start the breakout room. Maybe Guillaume, Enrique, if you want to add something.
But yeah, perhaps one thing I can add is that every project will have a specific design, so it's hard to have an approach which will work in all cases. And of course, when we work with Protist, so most of you work with Foraminifera, so they only want to have Foraminiferas. But when you work with Protist, you generally use general primers, and that's it. But if you are interested in a particular Protist groups in that bunch of organisms covered by the general primers, it's really worth to go to special to target the special groups, because you will definitely lose resolution. For example, if you're only interested in ciliates, then go for specific ciliate primers. If you're only interested for foraminiferas, go for specific foraminifera primers, because you will target a potentially much larger diversity with your specific primers.
How is the knowledge based on effectiveness of various methods for different groups? Is there a lot of published material to learn from, or do you need to do trial and error experiments? So this will totally depend which groups you want to investigate. In some groups, you have more publication, but basically we are still in progress. It's still a work in progress. And there is still a lot of works to do, a kind of standardization of methods per groups. And we should try stuff and think, oh, this one is the better way to analyze our data. Yeah.
Actually, so to give examples, ciliates are relatively well studied, and the methods are well established. For the tested amoeba, it's more work in progress. The primers are not really, there's no real official best primers. I don't know for forams, and for bacteria and fungi, actually there is also the same debate. So most people are doing using the general primers for this bacteria and fungi. Even people working with insects, they have actually explored the number of publications on meta-barcoding those last years, because they realized they can also describe the species with meta-barcoding of soil environmental DNA, so or water DNA. So actually, for those groups, there's more people working on it, so the field is evolving much quicker. And there's much more studies done on how many replicates you need, how many replicated DNA extraction, how many replicate PCR you need to saturate the diversity of your habitat of interest, environment of interest. For protists, it's depends.
Okay.
Always depends the amount of people working on it. So basically on ciliates, that's the most advanced group for this technique. So we have the most data and knowledge, and the rest is following.
Okay, I can give you a bit more information on forums because there's a lot of forums people here. It's still a work in progress, and I saw with the survey that there is a lot of people that are interested in paleoecology. And it's true that there is some groups that are working with ancient DNA to try to have new proxies in cores. But for this kind of research, it's even more difficult to get clean data sets to answer this kind of question. So even with recent material, we still have a lot of open questions that need to be solved. So it's great to wanted to have new proxy for paleoecology, but there is still a lot of work to do before we will have reliable tools to apply in paleoecologic field of research. Yes.
Okay. Hi, everyone. Thank you, David, for nice presentation. So I have just general question about the sampling. Let's say I'm sampling a pond, let's say for forams or tested amoeba. So the deepness or the volume, like, I mean, these details, how I will decide what is the right sampling strategy. So how deep I should go to capture, to know that it is saturated and like kind of this is enough to get the whole diversity. Thank you.
Yeah, if you remember in Sphagnum peatland, we tried to concentrate the testate amoebae to cover the whole diversity by letting them sedimentate in the bottom. And for forams, depending on the quantity, maybe you should try to do the same. I know that for morphological investigation, they just sieve that 125 micrometers. So they look on the big fractions. But with eDNA, we extract DNA from the whole fraction. So that's also a kind of things we should consider when you are doing that. So it really depends on the concentration of your organism in your samples. So maybe Enrique can give you more information because you did some sediments with testate amoebae in lakes.
Yes. Yes. Well, basically we try to, the things that the sediments are very fluffy. So the density of testate amoebae there is quite big. Although if you look at them with a microscope, you don't see them all. But there is even in 0.5 grams, you can have quite a lot of diversity, but and that's how we started. But I think that also concentrating testate amoebae either by filtering or that would be the best solution. But I think that most people here are interested in global DNA assessment, and that's why David said you have to adapt to what you want. Because if you concentrate all the organisms like you would do for forams of Testate, so for big protists, yes, you will miss a lot of. I mean, most of the diversity will be filtered out.
Okay.
Just question about samples. What are you thinking about lyophilization? How it's impact samples before DNA extraction? Just your opinion or some facts?
For this specific question, I really don't know. Yeah, did you read some stuff or did you make some experiment with that? Or?
I mean, we doing all the time lyophilization because we need to take our samples from Indonesia, for example. You have no choice. Okay, you have choice, but sometimes, like now, you could not buy any chemicals.
Yeah, and but you did that for the morphology, not for eDNA.
eDNA, for eDNA.
Okay. So maybe Enrique, you have something?
Yeah, I guess that, for instance, because you are also interested a lot on testate amoebae, I don't know how far all species can exist, for instance, and especially in the tropical rainforest. There are some papers, by the way, by Feusner, who was working on ciliates, and he just could not recover. He just when he dried the samples from Venezuela and brought them to his lab in Austria, he just found a couple of ciliates that were like very, pest-like ciliates, those that are found everywhere. And he could not find any specialized ciliates. And then he went back to Venezuela and took this sample again. And then but did not dry them. And then he saw a huge diversity. So even in the ciliates, a lot of them cannot exist. So I would try to avoid that. I would transport them even wet in the luggage, or if this can be done.
Yeah,
or
not possible.
Not possible.
Luggage. You need to keep till you get all permission. It can take 1 year. It's like you need to do something with your samples. And I thought lyophilization by -80 freeze drying, it should be actually
it's why during the presentation I say the best practice is to extract as soon as possible, but of course it depends where your sample comes from. I know that in our lab we are working with a sample from Svalbard, so it's also difficult to obtain some materials that have not changed. So you should of course adapt your strategy to what you can do. It's always better to extract the DNA as soon as possible, but if it's not possible, you should, it's a kind of weakness of your study, but it's fine. It's still valuable to study this kind of data set. Yeah.
Actually, I have a reference from former colleagues in Germany. They've tested freeze drying compared to direct freezing, and sampled in the sample they compare freezing directly at -80 and freeze drying and keeping the samples at seven days at room temperature after freeze drying, and they show only a slight reduction in alpha diversity for bacteria, but they got really similar patterns for fungi. So the impact by freeze drying is really limited. You can. So the aim is to freeze dry and then to send them directly, and then you put them back in the freezer once arrive in the lab, and seems not to impact much.
Okay, can you send this paper, please?
I will put the DOI in the chat.
Thank you. Because we are doing like this, we just freeze dry and then keep it by -20 till transportation, because we try to keep all what we can.
And why not? Well, RNA later is not, or LifeGuard is not possible.
I could not buy it now. It was difficult, or it was 5 times as expensive as it's already in Germany.
Yeah. So it's very expensive. We used that during my PhD. That was great, but now I know it's very expensive, and you can just store so very small volume. And if you want to work with a DNA extraction kit of 10 grams, for example, you will not put 10 milliliters of LifeGuard per sample. It will just cost you too much. So yeah. So we got a question from Maria.
Hi, no, I just wanted to add that there's actually a recipe online where you can mix your own RNA later, and it's very, very cheap compared to the bought one, which is about, I think, 500 euros per 500 milliliter or so, and the other one is just a lot of salt, and it's very easy to mix. I can look it up and then maybe put the link in the chat.
Yeah, cool. Mm-hmm. Sure. No, because it's a crucial point, the storage of our samples. So yeah, no, that could be a high interest for everybody.
Yes.
I collected some samples and intended to do some molecular work as part of my PhD, but the samples were stored at -80 for quite a number of years. Does that matter, or, you know?
I would try anyway. I think that you know what David presented before, and we discussed a little bit, and you know there are some people. I think it's rather you know very microbiologist mentality, like, oh, you haven't done this in the design of experiment, then forget about your things, just throw them away. It's not worth anything, and this is not true. Basically, as long as we know what the biases are, I mean, I think I would try. Is it soil which is frozen at -80?
Yeah, and it's some like sphagnum vegetation.
Sphagnum.
The sphagnum, yeah.
Yeah, I think this can. I mean, a lot of the things will be preserved. The only thing is with sphagnum, and you will see with our database, is that the sphagnum, when you extract from sphagnum, you get, of course, a lot of sphagnum sequences. And so half of the sequences will be sphagnum, and then you have to deal with the other half. Then with the other half, well, if you have also some troubles with experimental design and so on, so I don't know if it would be worth, perhaps what we are doing in the, especially in the lab with all the group, is that we actually try to do a clone library before starting a big, you know, high throughput sequencing experiment. We try to do a clone library,
Right.
and then look at what comes out, and depending on that, we go forward, or we may rethink the experiment. So if you get only very little diversity or only one thing or so, maybe it's not that worth to keep on trying.
Yeah, thank you.
You're welcome.
I guess I was wondering if you could speak a little bit more about the DNA extraction kits. And, you know, you mentioned when you tested 10 grams versus the 0.5 gram kit, you got really different results. Is there, I know that there's not a consensus, but I'm wondering, you know, in our lab group, we're starting to do multiple extractions from the same sample. You know, so we have the 0.5 gram kit, and then we'll save that soil and then kind of re-extract it a couple times to see, but we haven't done any, you know, tests. So I'm just wondering, you know, if there's any take-home messages or, you know, things you could expand on.
Yeah, so this point is really crucial, and it depends on your targeted groups. And I know there is different way to extract your DNA. So here in our lab, it's just a test for rams. Basically, the idea is to see if 0.5 gram is enough or if we should use 10 grams. But our first preliminary results just show that it's totally different communities. So, and as you know, with this kind of kit, there is a kind of mystery of how it really works. You don't know, there is kind of, yeah, they don't want to give you all the things because, yeah, it's a kit. So yeah, sometimes it's very difficult, and it deserves a lot of tests, and all these tests should be done per groups. So yeah, still a lot of work to do. And I'm looking forward to read your publication if you test this approach. Yeah, it should be interesting. Yeah.
Hello, David. I wanted to ask if you have some recommendation for DNA extraction kit for marine samples. Are you using also the Mobio kits like for sphagnum or soil?
Okay, so hi, Quentin. Cool. So you for marine kits, yeah. So it will depend a bit what kind of marine sample do you want to extract. For example, here in Angers, we extract DNA from sediments core. So for this one, we use the Mobio kit because maybe it can remove this kind of inhibitors of PCR products. So we found that it would be a good idea to use this soils design kit. But now, if you want to extract DNA from the filter for plankton, maybe there is other kits that could be better adapted to this kind of material.
Okay, but for sediment, you can use the kit Mobio then.
I recommend to use this one because we got some cool results with this Mobio Kiagen. But we use also the fast prep kit of 10 grams. It's another brand. Why? Because during this pandemic, we had some issues to order the Kiagen kit. They just didn't produce this kit anymore because they produce other stuff for the COVID. And they just sent us an email, yeah, we will do this kit, but maybe next year. So it's why if you want to do an experiment, it's better to order your DNA extraction kit and your PCR reagent before starting your experiment. And once you have everything, you will avoid some issues about that. Yeah.
Good point. Thank you.
Yeah, you are welcome. Hey, Natalie.
Hey, David. Hey, Quentin, and all the others. Yeah, I got a related question actually. So, how do we decide which kit should we use? Do you then, like, would you suggest that you test different kits beforehand? Like, I found it difficult. Like, usually now in the lab, I just use what other people did before, and I'm like, I guess that's fine. But yeah, how should we handle that? Would you say still it's better to maybe test different kits on a subset of your samples? And
sometimes it's great to just follow the same way than before because you will have the same kind of data set and you can compare the results. So it's a way to do the things. But at one point, we should have a methodological paper that really tests the things. And then you can prove, well, I'm using this kit because it's better for this kind of groups. And we don't have this kind of paper. There is some, but not so much. So it's why right now, every groups, every people that working with eDNA do his own pipeline, own bioinformatics, own kit, own way to analyze the data. And when you want to do a meta-analysis, it's very, very difficult because you have a kind of patchiness of stuff and there is no standardization in our field of research. And at this point, it's fine because we are far away to really have a generalization of the methods. So we need more methodological papers, but this kind of paper are quite boring to do, I would say. So, but sure, that's something we should do. And with this Foraminifera things, when I look on these results and obtain so many differences between 0.5 grams and 10 grams kit, I was like, what the hell? What should we do now with these results? You extract totally different communities with both kits that should extract the same. So yeah, it's very difficult. So it's fine, but you should have a very, your whole experimental design should have the same methods. If you change in the middle, it's just a nightmare and you cannot compare your results after that.
yeah, okay. So we just need so much more people working on Protist. So that's good.
Yeah, but it's a start. We should start somewhere. I mean,
yeah. No, that makes sense. Cool. Thanks.
I got a question because I want to look at soil protists, but in connection with the protists, I also would like to look at microarthropods if possible, and at the fungal community as well as at nematodes. So I guess the DNA extraction should be or be done using the same kind of kit. But then when it comes to primers, because you, like in the presentation before, you said you have some kind of general primers you can use for protists. But if I also want to look at microarthropods or fungi and nematodes, do you think, or is there any kind of primer set I can use to analyze them all? Does it, in your opinion, make sense? Or should I use maybe a special set for the nematodes, a special set for the fungi, and a special set for the protists?
Okay, for the fungi, there's an ITS set which is used by all the mycologists, environmental mycologists, and the good thing with that is that it gives you a resolution that goes to the species level. The ITS for that, and this is, I mean, it's like there's a lot of people who have been working on that. Then for if you are interested in nematodes, I would say, I'm not a specialist, but I would say that the V4, even the V4 primers, so the commonly used primers, will give you at least some level of definition, unless you're really interested in into one special group of nematodes, and then you might be looking at, for instance, CO1, the cytochrome oxidase gene, which is a gene that is used also in well, for entomologists and even people working on animals. This is like the universal barcode gene, so called. And then for microarthropods, I would say it's rather the same. Although in some groups, the 18S doesn't have a very good resolution, especially well, these are not arthropods, but rotifers, for instance. The resolution of the primers is not good at all. You have to go to other genes. But I would say, in general, I think nobody will criticize you if you use general eukaryotic primers for arthropods and nematodes. For the fungi, if you really want to go deep into fungi, you would use ITS. With the relative problem is that all the early branching groups of fungi will not come out, I think, that easily with the specific primers. You know, the chytrids and all these guys. But I don't know if you will find a lot of them anyway. So, yeah. Then it's the way you sample, of course. And for sampling, I would say for sampling nematodes, for sampling microarthropods, I would try to concentrate them, or to there are some specific traps that you can use there. And nematodes, you would take bigger amounts.
Yeah, I just want to see what's in the soil before trapping them, like around certain plants which carry a disease and other plants which don't carry a disease.
Okay.
So it's a bit of like the rhizosphere, close by bulk soil. Yeah, but you would do different PCRs for fungi and nematodes, arthropods together, and then using different primers again for the protist community.
Protists, if you want to look at all the protists, well, the best is to use these eukaryotic general primers. Like for, well, you have seen that there are two regions of the SSU that are targeted. Either is the V4, which is a larger region with which gives you, it provides you better phylogenetic information, so you can easily, well, relatively easily build some small trees, and they really look like how they should. And then you have the V9, which is way shorter, so you can have many more reads. And the V9, the good point is that the flanking regions are very conserved, so you would catch a bigger diversity. And so it's a kind of trade-off. It really depends. Maybe in your case, I think the V4 should be okay.
So I know it's possible to combine metabarcoding with stable isotope probing. Is there any major differences in methods and experiment planning besides obviously adding a tracer? Do you know?
I wouldn't know. I think we did one experiment like this with I don't know if you know Alexandre Jousset, but it was really long years ago, and we didn't have really the means to deal with all that phylogenetic information. And but it seems that just by separating heavy and light DNA and sequencing both fractions independently, you should be able to get them. I think it really works.
Yeah.
It's really nice. It's very nice to reconstruct actually food webs and so. So yeah, just do it. It's great. And tell me about it.
Yeah. So another things. So I know that they say with the whole sequencing that it's like per sample, like it's relatively cheap or cheaper
Yeah.
to do it. But I also know that to get sort of a good minimum data set, you often have. So how I understand it, if you send away a library for sequencing, I guess actually I'd ask the other group, but I thought I'd ask here as well, that you sort of pay for a full suite with a number, like with a certain amount of samples, I think, usually.
Yeah.
Right?
Well, what you do is a run. You do a run. So you just imagine yourself doing the PCRs. When you finish the PCRs, you pull all the products together in equal amounts in a single tube. And then you have this single tube, which is worth all your efforts for, I don't know, the past months. And you submit this to your sequencing company, and you hope that nobody will kind of mishandle it and will then, but everything is pulled inside. And the way you separate, you can only separate the sequences by sample, just only by bioinformatics. So because you have this kind of identifying sequences on each part of the sequence, which tell you, oh, this sequence, these tags, they're called tags. That's what David said with tag jumping. So you have this tag, I have this tag, so this means this goes to this sample.
Yeah.
And so what you do is that you send all your samples to do a run, like a MySec run or whatever. And the MySec run provides you, I don't know how many million samples, a million sequences. And then your total amount of sequences will be divided. You will divide your million sequence between each sample.
Yeah. So how I understand it, it's like per sample, it's relatively cheap, but because you are pulling all these samples, like the total run is still quite expensive.
Yeah.
So do you know of any efforts, or if this is at all possible, of just different like projects even pooling samples to send it away?
Totally, that's what we do, because otherwise we wouldn't have we wouldn't be able to pay that.
Yeah.
For instance, to tell you something that we have done and it works, is that we had a sampling design for all eukaryotes, and then we had some specific primers for tested amoeba, actually. And because you expect thousands of reads, of different reads of eukaryotes, and you expect just a few species of tested amoeba in your sample, what we did is that we put like 100,000 more DNA for the whole eukaryotes than for the tested amoeba, and it worked. So we got like several hundreds of sequences per sample of tested amoeba, and tens of thousands of samples for the whole eukaryotes. So it works. You can really play with that. It works.
All right. Did you ever hear of even like different research groups or institutes?
Yeah, you can even think about that. You can multiplex a lot. I think. I don't know what's the limit that you can reach. So if everybody is sending kind of 500 samples, I don't know how far this is possible. But without reaching the extremes, I think that you can pretty much do a lot.
Thank you. That's good to know.
Good morning, everyone. Yes, I have a question actually about budgeting in general, and it's such that we mostly do basic research, but we have many colleagues, as was pointed out. Everyone wants to do metabarcoding these days, and we get a lot of requests for collaborations, which is really nice. But the question they always ask us is, how much does it cost per sample? And this is like, for us, this is like asking, how much does the fish weigh? It's a very difficult question to answer, and we find that perhaps, especially as was pointed out, methods are evolving, and the way that you, let's say, replicate and process and all of these things is very much a moving target. So I was wanting to ask the community if anyone has kind of, let's say, a quick fix into how to, let's say, approach putting together a budget in a quick and dirty way. Or let's say, do you say per 8 samples, per 100 samples? And I mean, we're perfectly happy with adding on overhead to make sure that we don't work for free. But I just find it's a very difficult question to answer, this price per sample. I didn't know if anyone has any thoughts on that. I would be very grateful for feedback.
But to give you an example, one of the, well, maybe that's the most difficult to find, maybe the trickiest to find is the budget for the sequencing. So this would be like 2,000 euros, something like that. That's what I say roughly. Then I think that one flask of RNA later, and you cannot have the small version now. You have to buy a big one, and this is like over 1,000. You can just look at it. This is Qiagen. Qiagen is the company that does that. Then you have all the gels, all the PCR reagents, and all these things. I don't know by heart, but I would say like you have to count more than what it will cost you, of course, because you have to redo experiments. Then it's so basically, I don't know, something like 1,000, let's say. And then, of course, it depends on you how much you want to multiplex, or how many samples do you want to put in a single run, and all these things.
Okay, thank you very much.
You're welcome.
So I have a question for well, you know that I'm kind of the project time for writing, and the comparison of using the eDNA and the morphology, it's already been really used, and what do you think about these comparisons of the samplings? How it's kind of done in the bigger picture, kind of.
I think that this really depends on the group you're working with. So basically, because I think Anderson, you're working with Testate amoeba as well. So, well, Testate amoeba, you know, there is a lot of let's say cryptic diversity that is actually, or more or less cryptic diversity, so species that really look the same, but they are not the same, and they don't do the same in the environment. So in that case, if you're doing like just morphological observations, you would miss that part of the diversity. There is also a lot of very small Testate amoeba that we didn't, I mean, that just pass unnoticed, and all these organisms, they are, you just miss them if you're just counting, because they are transparent, because they're inconspicuous. For many reasons, you miss them. And but still, I think for the Testate amoeba, well, if you want to build a project, that's what I put in my project. So I put also that we need to document the cells, and we need to barcode more things, because there is a lot of Testate amoeba that we don't know, and a lot of diversity that has never been barcoded. So if we don't have a good reference database, we are in a, we just don't know. We have sequences, and these sequences mean nothing if you don't have if you don't have a reference to. And it's also the way to link what you can get with DNA and modern methods, 21st century methods, with what has been done before by, you know, people like Bonnet, like even Penard, or all these people, and Deflandre, all these people. So I think it's very important to keep on doing microscopy also, not just DNA. And especially in your region, Brazil, I think the diversity has almost not been registered. And I don't know if you know BIAS, Bia, I don't remember her name now, her family name, but
Bia.
yeah, Bia. But so she did a guide of Brazilian tested amoeba, and we show you some things that you just say, wow, this comes from Mars. So there are many things, especially in tropical areas, that have never been seen, never been described. This is like it's the jungle there. I mean, and they are just as beautiful as the birds. I mean, tropical birds are very beautiful. Tropical tested amoeba are very beautiful too. So yeah, there's a lot of work to be done.
We will need to follow the same protocol if we want to compare between samples or whatever. But for example, we are going to try now, we are going to start with a methodological paper. And one of the things that we want to test is with a mock community in my culture, so normal culture with NMAS, and then inoculating this in soil and doing a DNA extraction. But yeah, what can I do, or what protocol can I follow to have the same protocol in soil and in NMAS or liquid culture to be comparable? Because yeah, I guess that if I use only one, it's going to be much better, or for soils or for NMAS. So yeah, in this kind of methodological aspect that we are trying to approach, I don't know if using only one that is the same for both will be the perfect solution. I don't know.
I'm not sure, because you compare, for example, if you are looking on plankton communities, so you will filter water. And you should, for example, did you read the Lara paper, Ocean, the first one, metabarcoding, the well-known one? They filter thousands of liters of liquids to obtain the communities. And now we are working with soils, and we extract DNA from 0.5 or 10 grams of soils. So look, there is a difference of magnitude of the thing. So, you can really compare this. It's an open question. And we try to compare soils, marine and freshwater, and we assume that each of the methods collects whole diversity. That's the whole point. You can compare the different environments as soon as you think you cover the whole diversity. That's my idea, but of course, we need more tests to really be sure that what you have is the real diversity.
Yeah.
And this question is really depending on the ecosystem you are studying. Because during my PhD, I studied sphagnum peatland, and it's a very stable thing. And now I'm working with littoral zone. And you collect a sample here, and you collect a sample here, and you have a different community, because it's totally moving and totally, and that's just crazy. So you should really adapt your way to sample the ecosystem depending on the dynamism of your ecosystem also. So yeah, still a lot of work to do with this.
Yeah, thank you very much.
Oh, you are welcome.
Do you have any advice on what not to do at all? Some silly mistakes not to do, or some obvious things to avoid?
My really great advice is for the field work and the laboratory work, you will find many publications. It's very easy, this step, I would say. If you design well your experiment, it's quite straightforward. But then to analyze the data, to do the bioinformatic parts and the statistics, there you can really do a lot of mistakes, because it's really a huge field of research. And I know that there is a lot of PhD student or master student, and the professor just say, oh, you should analyze this data set, without any advice on how to do the things well. And in this point, you will do a lot of mistakes, because there is a lot of specific points. If nobody told you what to do, you will do the things wrong. So yeah, if you want to start, my advice is collaborate with a group that do the things. Yeah, that's really my best advice. Don't work alone on this data set, because you will do all the mistakes that everybody is doing. And then people will say, oh, look, this is wrong because you forget to do these steps and correct your things because of that. So yeah, just collaborate with someone that know, or hire someone, and you will be able to save a lot of money and time. Yeah.
I may have another question if nobody has. Nowadays, there is the choice between OTUs and ASV.
Yeah.
Do you think one is better than the others? And if yes, in which case should we use rather one or the others? Do you have some advice?
Okay, the OTUs is operational taxonomic units. It's more the generic names. ASV, it's more related to the data2 clustering algorithm, or other ways that use this error model to build the ASV. So it's just the name of ASV versus OTUs. No, with Protist, we are using a lot of Swarm. It's a great algorithm, and they there is a V3 of Swarm that are out now, but I didn't test this this new version. Or we use also Data2 because a lot of people are using that. My advice is they should provide you quite the same kind of outputs. And Christophe Sepe did some experiment where he studied both algorithms, Swarm and Data2, and he has something really similar between the two outputs. So it's just a way of thinking. But as you know, the number of ASV OTU you will get is just depending on the parameters you put in your bioinformatics. So if you want 10,000 ASV, you will have 10,000 ASV, but that doesn't make any sense. So see, it's just the way you clusterize your data sets. But for me, it's better to use Swarm or Data2 than the clustering algorithms that are developed for bacteria, where you set up a threshold of 97%, because this 97% of diversion between ASV doesn't make any sense in Protist, because we have a barcoding gap between species that can be up to 6% or less than 1%, depending on the different cases. So you should have an algorithm that adapts the threshold depending on certain conditions. And Data2 and Swarm are really better than other algorithms that exist for bacteria.
Okay, so they are quite equivalent then.
They are quite equivalent, not exactly equivalent, but your main ASV OTUs should appear the same way.
Mm-hmm.
You shouldn't have a totally different output depending on your clustering. Yeah.
Do you think it would be wise to try both and compare, or just use one of them?
You can try to do both, yeah, of course. And you should have quite the same output. Swarm usually produce more OTUs than Data2. And that was one mistake that people didn't really understand when they read the Lara paper in 2015. They found like 200,000 OTUs. So in the newspaper, for people, they found 200,000 species. And it's not the case, because they just oversplit the species in many, many different OTUs. So it's why for some people, one OTUs or one ASV is one species, but it's not true.
Mm-hmm.
So we should really be careful. And we have this case with Foram, and it's a bit tricky to treat that because, yeah, some species have different variants in the sequence, and how you should pool them to have the real value of number of reads depending on the species. See? So, yeah. It really depends on your groups.
Yeah, I was wondering if you should we should explain the reason why are we using ASVs or OTUs in our papers, because I guess maybe some journals or some reviewers, they say to you, yeah, then why? And for Protist, why are we using one or the other? Because yeah, I can imagine that the concept of a species here in this big massive group is almost impossible to define. So what explanation can we give in a paper to say, yeah, I'm using ASVs because, not because I want, but because of some scientific explanation?
Yeah, you can use both, and if the reviewer complain about that, you can still do the analysis with a swarm. And it's not a lot of work. If all your scripts are ready, you can do that and put as supplementary material, and you should really obtain the same kind of output, because all your main OTUs and ASV will be the same. If you don't have the same, maybe you have an issue with your data set, but usually you will find exactly the same kind of big groups, and the big pictures is the same. It's just, it's not the same way of thinking to the clusterization. And the main thing is with OTUs, you will a bit oversplit your community, and you will generate a bit more diversity, genetic diversity than ASV. So it's why now I would say people prefer to use this data2 algorithm because they generate a bit less, and it seems a bit better for Protist. But there is still this swarm algorithm, and there is a version 3 that are now out, but I didn't test this version, so I don't know if it resolves some issue or not. I should test this one. Enrique, did you?
According to the one who did it, the swarm 3 resolved the issues which were resolved by data2.
Okay, so it should be more or less comparable now.
Yeah. And but it's always better to use the latest version because it's the fanciest one. You know, I think most of the people started using ASV just because it was fashion to do so.
David, do you hear me?
Franz, I can hear you, yeah.
Maybe you can give Mathieu some examples. You have been here in our lab now for almost a year and a half, and you know that we have tried to develop eDNA independently without really knowing how to do this. So like the television program Nightmare in the Kitchen, Cauchemar en Cuisine, what horrible mistakes did you find out happened in our lab? That would be a few nice examples.
Do you want that I can explain and give you some example? Yeah, of course. Okay. So the first things when you analyze your eDNA dataset is something very simple. You will obtain some ASV, OTUs, and per sample you have different number of reads, right? And the number of reads you get from the machine is just depending on the quantity of DNA you will have in input. So you put more DNA in the machine, you will have more reads. That makes sense. And so at the end, normally, if you pool with exactly the same quantity of DNA, you will obtain exactly the same quantity of reads. So when you analyze your data set and you see a correlation between the number of reads and the number of ASV or OTU, what does it mean? It means the more sequence you have, the more ASV you have. But it's just an artifact of sequencing. It's not the reality, this correlation. So you should absolutely test this fact. And if you get this correlation, you should remove this correlation. It's why normally we do a rarefaction of your reads to remove this kind of artifact. But rarefaction, there is some publications that people don't like too much because sometimes you remove too much reads and you lose some information. So yeah, that's the kind of things. If you don't know that you should do a rarefaction and you have this kind of positive correlation between the number of reads and number of ASV, all your conclusion in ecology will be potentially be wrong. And I read some paper where they did this kind of mistakes. So but if nobody told you you should do that, oh, you will discover that it's very important. So it's why it's very crucial to work with people that are that know what to do with this kind of data sets. Yeah. And that I realized in a master defense that the master students did all this analysis before checking this correlation. And I told him, yeah, I'm sorry, but maybe all your results are wrong. And we test, and indeed he has this correlation. So he had to change everything. So that's something you should be aware of.
What about the intercomparability between datasets in Swarm versus data? I know that it's pretty easy with data, that you don't have to do the whole pipeline again, but I haven't done Swarm yet so far. Is this really an issue with Swarm, that if you have two sequencing runs and you want to compare them, that you have to start from the beginning and put them in the pipeline together?
Yeah, the main issue is it's not the same input from both algorithms. Meaning for data2, you keep the R1 and R2 separated before the clustering. For Swarm, you should merge R1 and R2 before running Swarm. So it's not a big deal to do that, but it's a bit different the way you obtain the things. Yeah.
Okay, thanks.
For example, tomorrow I will explain data2, because for data2 it's an R script, so it's not with a Linux shell. So we will begin to start with Linux to do the preparation of your sample, do the clustering with R, and the assignation will be done with Linux again tomorrow. It's just to show you that you should adapt different tools you want. Yeah.
Okay, thanks.
And about changing and stuff, there is a huge study that will once be out, I hope. We provide the team of Colombo and Fred May and all these people from Tara Ocean, all our data set of V4. And they have just merged all the data sets together to obtain a huge things. But there is like more than 1,000 data sets. So it's very massive. And they wanted to compare the whole V4 region. But they have noticed that if to target the V4 region, you have different kind of primers for the V4. And you have like 10 or 15 different couple of primers. And depending on the couple of primers you will use, you will have different outputs. So to compare a V4 data set, depending on the primers, can change a bit. But still, it's an ongoing project, and I'm really looking forward to see what's going on with this one, because it will be huge. Yeah.
Yeah, if I can, another question related with this. Do we have to select depending if it's a short read sequencing or a long read sequencing? Do you think the ASV OTUs should change in that way, or it should maintain the same? So it doesn't mean anything if we have a short read sequencing or a long read sequencing.
You mean long or short reads?
Yeah, so if we have short or long reads, do we have to switch to ASV or OTUs depending on that, or the type of sequencing that we are having?
No, I think it works the same. No, I don't have any issue with short read or long reads. But one thing that you should keep in mind is if you have a huge variation in terms of sequencing length, meaning if you have very short ASV and long ASV, it means when you sequence that, the short one will be really more sequenced than the bigger one. You will have a bias. And in some case, it's very an issue. For example, with forams, because I'm working with this group now, we observed that in our sample, we have a lot of monotalamides with eDNA. This group is only very short reads, and the other one have very longer reads, but they are less present in our sample. But it's maybe due just to a methodological, technical things, artifacts. So when you interpret your diversity based on the number of reads, you should be careful of this aspect also. It's why I say it's only a semi-quantitative data, the number of reads, and for some scientists, even it's not good enough, and you should just work with presence-absence matrices. So it really depends what kind of data set you get, and the interpretation of the number of reads, it depends on many different factors.
Okay, thank you very much.
Don't be shy.
Just have another question, but it's more bioinformatic. Do you think it changed something to have a Linux terminal or a Mac terminal, or it's exactly the same?
No, it's based on a Unix core, so it should be the same. When I wrote the PDF with the requirement, I just say I didn't test that on the Mac, but it should work.
Okay.
But you know, sometimes there is strange things that appear between the different OS. So yeah, but you should adapt stuff and it should work. Yeah. Okay. Tomorrow, for example, I will present you some tools to transform a text file. And if you are on Windows, you have an extra step to do, because on Windows, they generate at the end of your line some hidden characters that you should remove to do your bioinformatics. And these things doesn't exist in Linux, and I don't know in Mac. So depending on the OS, you cannot use exactly the same way, because there is this kind of small errors that can appear every time you are doing your bioinformatics. So yeah.
Okay.
I have a question about what you said earlier about the Foraminifera diversity. So I have very little experience, but I noticed that Foraminifera has like huge diversity. So if you are looking at regions like V9, you can see like very low taxonomical level of identity. So maybe if you're using this kind of regions, you will not see the diversity in groups like Foraminifera, not as deeply as other groups.
Yeah. So for the graphs that I presented between the 10 grams and 0.5 grams, for this one, we use a specific marker for Foraminifera. So this marker is designed in the 18S, but really to focus on Foram, it was designed by Jan Pawlowski. Well, that's a guy that really work with eDNA and Forams. So in order to really obtain more diversity, the real diversity of these forums. But there is many issues with this specific marker and eDNA because there is a short read. So for some groups, some ASV have only 60 nucleotide long. And for some groups, you have 180 nucleotide long. So you have some ASVs that are 3 times longer than the shortest one. And that means when you sequence them, you will sequence more the short read than the long read. So you will have some bias with that. And typically with this marker, the short read are the monotalamids. And when you see some presentation using eDNA and Forams, one of the common observation is we found a lot of monotalamids in our data sets. But potentially, it's only an artifact of sequencing. So you should be careful when you interpret your data with this.
You talked earlier about the extraction of DNA and the problem of the shells. Did you try to dissolve the shell, like with eDTA or something like this?
No, I didn't try to dissolve the shell because I play in my eDNA experiment, I was interested by Foram, but also by the total diversity of eukaryotes. But once you extract DNA, you can absolutely test different marker on your DNA extraction, and it should work. So I just respect the protocol given by the extraction DNA kits. And sometimes it's a bit easier to publish your data if you say, yeah, I did according to the manufacturer's instruction. But maybe, yeah, for Foram, that's an open question. And in the future, I would like to do some more tests to really select the best DNA extraction for Foram. I was wondering how you can build a reference database, or where to find information on building a reference database for Protist. I work in remote environments, so often reference sequences are scarce. I know how to do this with animals, but I have no experience with unicellular organisms. Are there large differences? So for this question, what I can answer is we use for Protist, we use PR2. It's a big database for the 18S. So it's a great database for the whole diversity of eukaryotes. And it was a curated reference database. So the people that put the sequence there, they just check that everything is okay. So it's a great to have the overall diversity. But then if you want to study specific groups, you should build your own reference database. For example, with Foram, I noticed that PR2 is not really well adapted because they lack a lot of sequence that exist. So to do that, you can go to GenBank and search your group of interest and download the different sequences. But so I would say it depends of your groups. And for example, with Foram, I download like 80,000 different sequences, but then you need to clean this sequence. So remove the sequence that are not related to your marker. So all the transcriptomics stuff you should remove. And then you should remove all the duplicate sequence because you have a lot of duplication in GenBank. And then you can really remove everything that are not related to what you want. And at the end for Foram, I have like 3,000 reference sequences in my database, but it takes a lot of time to do that for the groups. But you are lucky if you work with a group that have so many sequences, because if you use specific primers, maybe you will have very few sequences as reference. And that's the real issue. Because if you want to increase the value of your database, you need to sequence single cell to obtain the barcode of your specific species.
I do have one more question. If you have, because you were also speaking about sampling and how many replicates and this kind of stuff, so I'm going a bit in another direction, but I typically work with pot experiments where then I have my experiment and my replication, and I was always wondering how many replicates should I have for the DNA extraction. I mean, then I also have, of course, for plant properties and stuff, I also need a certain amount of replicates, but yeah. I think it's always specific and we never really know how many we should have, but do you have any opinion on that?
Okay, so basically for you, it's your experiment is very well set up. It's mesocosm, right? Or yeah. So for that, my advice is work with a statistician. I'm sure in your university you have this kind of crazy guys that work with maths and stuff, and they can really provide you this kind of information because they are used to that. But then it's true when you are speaking about biodiversity, to cover all the diversity and this kind of question, it's a bit more tricky. And you don't know exactly how many species do you have and all these kind of things. So my advice is try to speak with a statistician. He will give you a number, but it will not be possible because it will say, oh, we need 15 samples per mesocosm. And you say, no, come on. But it will give you a general idea according to your experimental design. And then you can think, oh, okay, realistically, what should I do? And then it's fine. It depends on the level where you want to study your things with your mesocosm. Because for me, I went crazy when I see some diversity plots, when you have some box plot with only 3 points. And I reviewed some paper with that. They present box plot, but there is 3 points in the box plot. That makes no sense. So you should adapt your statistics depending on the level you are working on. If you pull your different mesocosms together, you will have much more sample, and you can do your box plots. But if you treat all your things separately, you will need much more samples if you go this way. See? So it depends on the orientation of your questions, I would say.
Well, I guess it is so. It depends a bit on the. So if I have a treatment, for instance, on my pot, and if I expect, I kind of expect the same effect to happen on my community there, and then if the effect is really similar everywhere, then these communities, the way they change should be similar, and then I should not have too much differences in my replicates, and then maybe not need so many replicates, right? That's also something to consider, I guess.
Yeah, but for some authors, they just combine the replicates. Before doing the, see, you have your samples, and you have maybe 3 replicates, and they say to have the whole community, I just pull the 3, and that's 1 replicates of my mesocosm. And then I compare this one to the other one.
To the other, yeah.
It's another way to do the things. So you can keep your replicates separate, or you can try to pull them also. I saw both strategy.
Yeah, okay. That makes sense, I guess. Yeah, it's always the same question, I think, in any experiment anyway. So
it's always the same question, and you should find the right threshold, because, of course, I would say take as much as you can, but it's just not possible because it's quite expensive, and the time to extract all these things and do the lab work and things. If you have 1,000 samples, you will just die. So yeah, just try to find the right threshold between what you expect and in which level you want to work. Yeah.
Okay. Yeah. Thanks. And just coming back still on that, because you gave the example of a very stable environment and a non-stable environment. How did you handle that? How many replicates did you decide to take in your case?
During my PhD, we took triplicates per microhabitats, and during my postdoc here, they took not enough replicates. It's why now we have some results, and it's very difficult to interpret them. We will be able to say something. As I said, even if it's not collected the best way, you still have some value in it. But sometimes you say, okay, but I have this tool, it's from the same environment, but it's totally different. Why? It should be the same, but no, it's not, because there is this issue with it's not very, it's too much heterogeneous. So yeah, we should deal with that. But in the future, maybe we will plan to have less sites, but much more replicates. And so you should adapt your strategy, and that's the main point. And in this 100 people in this conference, there is people working with different kind of environments, and each environment has its own rules. So I cannot say, oh, for this environment, you should take 3 samples. Oh, that makes no sense. So yeah.
Yeah, true. Thanks.
Yeah, you're welcome. Alex, anything else?
Yeah, more into the laboratory work, and now taking advantage that Natalie is here. Yeah, I have a doubt in terms of culture, because when I try to create some culture from soils, I'm getting almost the same with the NMAS protocol. Especially, I'm not actually obtaining almost any tested amoeba. So I don't know if some of you, especially Natalie, that I know that you work with that, did you use more than one method or protocol? Or yeah, I don't know, because actually I don't have any tested amoeba.
Which marker did you use for tested amoeba?
No, I mean, I'm just taking cultures or creating cultures from soil.
Okay, so if you want to do cultures for tested amoeba, sometimes it's very tricky. Enrique can give you much more information about that, because there is a
tested amoeba cultures.
Yeah.
But I think, well, I don't know. In your case, I think the only one that will grow, I think, would be Cryptodiflugia.
Yeah.
I think that's the only one you can hope to have. Then you have some Euglyphas, but even to get Euglyphas, you in a 96 well plate, they wouldn't really thrive.
Actually, I have one culture of Cryptodiflugia, but I don't know how did they obtain it, because I have done.
Send.
I know, I'm with you. Yeah, but I don't know how Stefan. I don't know if it was Stefan or you, Natalie. I don't know how did you obtain this one, because I have tried several times and I didn't get any single tested amoeba. And some things look like a shell, but still I think it's completely dead because there's nothing inside. So
yeah, I think it's Stefan. Stefan got it, I think, but I don't know how he did it.
Then I will ask him, yeah, because yeah, I always obtain the same flagellates, the same amoeba, but yeah, I want some diversity also.
Where do you look at?
Where, in which
what
sense?
What type of environment are you looking at?
Soils, from soils, everything from soils. So actually,
yeah, what soil? What soil?
The ones that we are taking actually is quite random. Like maybe I have a walk and then I decide to take a sample. So I have
garden soil. This tested amoeba you would be luckier if you try the forest soil.
I have also with the forest soil, but with all the organic material that is in the forest, it's super difficult to have something clean from there.
Yeah.
Yeah, also. I'm having a sort of a mix of everything here around Wageningen to, yes.
And then you can do serial dilutions. And taking your soil, you do several dilutions, and I think that's the best way to get a kind of Cryptodiflugia. But you know that tested amoeba, I work on tested amoeba, but there are not a majority of what you can find. I mean, numerically, they are not very abundant.
No.
They're big, so they eat a lot, and they really play a role in the food webs.
That's the point. I want to use it in an ecological experiment, and I would love to use one of those in terms of ecological role.
Yeah, because
that's exactly the point for this species, and for Foram it's the same. If you want to do ecological things, you should have cultures of them, and it's sometimes very difficult to obtain one single cultures.
Most of the time it's very difficult, but you have species that grow pretty well. I mean, there are cellas.
Yeah, but
aquatic are cellas grow well.
Yeah, but there is some. But in comparison with total diversity, it's just like bacteria. You can grow some, but and for us, if we want to go forward with these ecological questions, we really need to have these cultures to make some tests of ecological niches and all these kind of things, and we still don't have.
Euglyphas would be a good something a little bit intermediate that is not a Cryptodiflugia, but Euglyphas would feed also on larger particles, and it could play a role in the food web also. That could be an option for you.
Yeah, but I don't know if Ralph Meisterfeld gave all his information about how to culture stuff, but
no, Euglyphas are. I mean, you can order them.
I mean, yeah.